We will proceed now to the year in review. This is a session we started about 10 years ago where we invite 4 experts in different areas of breast cancer research to give us an update of the highlights of the year on the areas of basic science, translational research, early breast cancer, and advanced breast cancer. So we will have 25-minute talks back to back. I'll introduce the first 2, and Dr. Virginia Kaklamani, my co-chair, will introduce the next 2. There'll be no questions. And the first one is Dr. Xiang Zhang from the Department of Molecular Cell Developmental Biology at Baylor School of Medicine. And he will give us the year in review on basic science in breast cancer research. Dr. Zhang.
I would like first to thank the planning committee for the invitation. It's really my distinct pleasure and honor to deliver this year in review basic science talk. I have no disclosure to make. So okay. There were 25,000 papers published in the year of 2021, according to PubMed. So I took 2 approaches to identify the topics that may be of general interest to the audience. Using an unbiased approach, I analyzed the keywords that frequently appeared in the titles of these papers, and there 4 really stood out. These are models, immune, metastasis, and resistance. I also took a biased approach based on my own reading and interpretation of the literature. I decided to also cover metabolism in this talk. Indeed, if you look into the abstract of this paper, there's 18% of breast cancer-related papers have metabolism or mitochondria in their abstracts. I also would like to discuss tumor heterogeneity and plasticity, as well as diversity of tumor microenvironment. And this is because we have learned a lot in the past year because the increase of application of single-cell omics. Accordingly, I divided my talk into several sections, focusing on tumor heterogeneity and plasticity, metabolism, immune, tumor microenvironment diversity. And these 4 are interrelated, and they all play important roles in metastasis and resistance. And also, finally, I'd like to discuss experimental models, as well as without preclinical models, we cannot do great science. I would like first also to apologize to our colleagues whose fantastic work cannot be covered in this 25-minute talk. So let's first look at heterogeneity and plasticity of cancer cells. By discussing 2 papers, I'd like to pass the message that not every cancer cell is born equal, and not every cancer cell will stay the same. So the first paper is from Dr. Nick Navin's lab. The authors performed single-cell DNA sequencing on over 16,000 cells from 8 triple-negative breast cancers and cell lines. They discovered a large number of subclones, which are organized into a few major superclones. So there's a very complicated clonal structures. And they deduced this has probably happened by first have a p53 loss, and then followed by transient genomic instability that create a large number of copy variations. However, this is not the end of story, as new aberrations continue to develop. And over the course of evolution, there's no single or few clones that become dominant. And this really suggests that tumors are constantly altering and trying to explore the genomic and the phenotypic fitness space. And therefore, this subclonal diversity is dynamically maintained. So not only cancer cells are heterogeneous, not only at the genetic level, they're heterogeneous, at a phenotypic level, they're also very heterogeneous. And I'd like to discuss this concept of phenotypic plasticity, but without talking about epithelial mesenchymal transition, and this concept will not make sense. So what is EMT? EMT refers to the process of cancer cells lose epithelial traits and gain mesenchymal traits. And this has been thought essential for metastasis, as the mesenchymal traits really renders cancer cell migratory and stem-like. And Dr. Yibing Kang has written a nice review several years ago summarizing the pathways that can activate EMT. However, during the recent years, there's increasing evidence suggests that it's not the mesenchymal trait that's important for metastasis, but rather it is the ability of a cancer cell to change, to switch back and forth between these different status that make them metastatic. And in this past year, Dr. Robert Weinberg's lab published a review summarizing the more recent evidence we got from research. And he summarizes that cancer cell can leave primary tumor not only in the mesenchymal status, but also they can leave without completely losing mesenchymal epithelial traits. And the different cells can stick together to form CDC clusters. And there are cancer cells that exhibit hybrid or quasi-mesenchymal phenotype, and so-called quasi-mesenchymal tumor cell. And it is indeed the CDC clusters and quasi-mesenchymal tumor cell that possess the highest metastatic capacity. So along this line, Dr. Sindhu Ramani's lab performed the research in-depth characterized intermediate EMT status using MCF10A cells exposed to TGF-beta treatment. So at different time points of experiment, they exposed these cancer cells to single-cell RNA-seq, and they identified several intermediate cell states. And they characterized the pathways, gene signatures associated with this different intermediate state, and used them to interrogate human clinical data. And indeed, several of these signatures have poor prognosis in human, supporting the idea that the cells with hybrid or partial EMT status are more likely to drive the tumor progression. So to conclude this session, we learned that genetic heterogeneity of breast cancer is dynamically maintained throughout the tumor evolution. And the ability to change the phenotype, plasticity, is an important feature of a cancer cell to see the metastasis and drive resistance. But this also opens new questions. How do we integrate these two levels of heterogeneity together to understand metastasis and the therapeutic resistance? And this really requires us to have an approach to simultaneously assess cancer cell genetic information as well as phenotypic information at the single cell resolution. So not only cancer cells are heterogeneous inside of a tumor, microenvironment is also heterogeneous. And different patients may have different tumor microenvironment developed, as we have heard from Dr. Carey's talk earlier this afternoon. I would like to discuss two papers along this line. The first is from Dr. Aaron Newman's lab. They use large-scale bioinformatic analysis. They collect a large source of data, including 16 types of solid cancer that also include breast cancer, and use them to input a bioinformatic algorithm called ecotyper. And ecotyper deconvolute the data, identify different cell lineages, as well as the same cell lineage but at different functional status. By looking at this data, they find that really each tumor is not a random mixture of different cell type, different cell status. There's a co-occurrence of different patterns, which they refer to as ecosystem. I think this is a great word because it reflects that there's a co-evolution between different cell type and new tumor cells. Ecosystems obviously indicate distinct cell-cell interactions, and they also correlated to prognosis. The second paper, Dr. Alexander Swarbrick's group focused more on breast cancer using a technology called SABCS. So this technology simultaneously profiles surface protein as well as single-cell transcriptome that provide high-resolution immune cell profiles. They apply this to over 20 breast tumors. Again, identify different cell lineages as well as the same cell lineage but different status. And ecosystem were defined similarly according to the recurrent patterns of different combinations of cells and their functional status. And again, this ecosystem is correlated with patient survival independent of the known parameters. So both papers tell us the tumor microenvironment varies from patient to patient, and there's a specific role of particular cell type and they need to be understood in their ecosystem because there's a co-evolution history. And for this particular process, such as immunosuppression, different tumors may utilize different mechanisms. But how do tumors evolve this different ecosystem? Is there intrinsic property that initiate this whole difference, or there's a combination of tumor intrinsic as well as host factors? And how do different ecosystems really impact therapies? This warrants further research in the future. So let's turn our attention to metabolism. Metabolism may play multiple roles in tumor progression, as we will see from the following 4 papers. The first is from Dr. Ramakaka's lab in Canada. So these authors profiled metabolome of mammary epithelial cells. As we know, mammary epithelial cells are not homogeneous. They differentiated from mammary stem cells, and they're organized into 2 lineages, the luminal lineage and the basal lineage. And this lineage of cells exhibit different metabolic profile. Basal cell enrich glycolysis, whereas luminal progenitors enrich oxidative phosphorylation. Their mitochondria also appear to be different, both functionally and morphologically. And very importantly, hypothetically, these different epithelial cells are precursors of different subtype breast cancer. If you look at the corresponding subtype, these features retain. Going one step further, Dr. Zhiming Shao's lab in Fudan University in China directly profiled triple-negative breast cancer at multiple levels. They studied 465 triple-negative breast cancer, and looking at their metabolic profile, they defined 3 metabolic pathway-based subtypes. So this classification shares some information with the existing subtype in the literature, but also has some distinct information in there. They exhibit different therapeutic responses. MPS 1 is more sensitive to glycolysis inhibition, whereas MPS 2 is more sensitive to inhibition to lactase dehydrogenase in combination with checkpoint blockade. So let's go even further to metastasis step. So Dr. Matthew van der Hadden's lab demonstrated that in brain metastasis, there seemed to be a requirement for fatty acid synthesis. So they used HER2-positive breast cancer models, and in brain metastasis of these tumors, they discovered longer fatty acid chain. So that suggests increased fatty acid synthesis activities. And this may be attributed to increased availability of lipid in brain tissue, a unique metabolic environment. And very importantly, they show that if you target this synthesis process, you can selectively inhibit brain metastasis. So this highlights the possibility that cancer cells need to adapt to the local metabolic environment during metastasis that render them vulnerable to certain therapies. The last paper in this session will focus on a cell biology-oriented study, really linking a very basic process to metabolism. This is migration and invasion. As we know, the matrix differing and the high mechanical load can stabilize microtube. And this is through glutamylation of the microtube. And therefore, glutamine catabolism is necessary in this process. And there's outside-in process from mechanic force that can activate glutamine catabolism, and the inhibition of this process can inhibit metastasis. So to summarize this session, we learned that metabolic features contribute to define tumor heterogeneity. So this is another layer of tumor heterogeneity. Adaptation to metastatic environment may bring up novel therapeutic targets. And important biological process such as migration invasion may actually be regulated by metabolism, which is underappreciated before. But how do varying metabolic environments in different organs contribute to metastasis? Are there similar mechanisms parallel in other organs? So in the end, maybe metastatic organotropism is partly determined by this different metabolic environment. And this is an open question. And going forward, we still need to think about how to integrate this information into our precision medicine research. So now let's talk about immune system, how the immune system can be leveraged to fight breast cancer. And this may not be a question with a consensus answer to it. So I formulate the question as, should we prioritize research in tumor immunology in breast cancer? And many people may not be very optimistic because most breast cancers are immunologically cold. And also, as we have heard this afternoon as well, that the checkpoint blockade therapy only achieved a small, I would say limited effect in the triple-negative breast cancer, and some have severe side effects. But many other people, including myself, are very hopeful still because the immune cell infiltration is prognostic of survival therapeutic response. Also, many chemotherapies, targeted therapies that are not designed to target the immune system actually works through activating of immune response. So that we will come across a few examples in this session. And finally, it is my personal opinion that the evolvability and the versatility of our immune system really make it unique and perhaps the most powerful weapon to outcompete tumors, which are also evolvable and versatile. It's only we need to help our immune system appropriately, identify how they are crippled in fighting the tumor. The first paper we're going to discuss in this section is coming from Dr. Lana Welm's group. Dr. Welm's group have done a series of work in the past, characterized the RON kinase role in metastasis. And RON kinase really exists in multiple isoforms. There's a full length and there's a short isoform. In this particular study, Dr. Welm's group showed that knockout of short form RON can significantly reduce lung metastasis in preclinical models. And interestingly, and this is happening because T cells are recruited to metastasis. And if you knock out T cells, this efficacy, this effect is abolished. And similar to genetic knockout, pharmacological inhibition also achieve the same effect. So this highlights a scenario that by targeting a kinase, you actually works through activation of the immune system. Another study from Dr. Mikala Ajiblad's lab showed that you can actually turn a hostile immune microenvironment really into a hospitable environment. And this is achieved through two different things. One is called monophospholipid A, which is a LPS derivative, and the other is interferon gamma. And these two things turn the immunosuppressive macrophage into tumoricidal macrophages, turn the bad macrophage to good macrophage. And the good macrophage in turn secrete IL-12 and TNF-alpha, which activates cytotoxic T cells. And you can inject these two things to the tumor and reduce tumor progression and the metastasis. The third paper in this section is done by Dr. Jennifer Griori's lab. They focus on BRCA1-associated tumor, and they show that BRCA1-associated triple-negative breast cancer has increased infiltration of T cell and macrophage at the beginning. And they respond to PARP inhibitor, but resistance can be developed due to the accumulation of macrophages. If you eliminate macrophage by using this anti-CSF1R treatment, you can overcome the resistance, so the two treatments actually synergize. But again, this is dependent on T cells. If you knock out T cells, this synergy is gone. This is another example that therapies that are not intended to target the immune system actually works through activation of the immune system. Let's then switch a little bit to talk about distinct stage of metastasis, which is dormancy. Dr. Momo Bentay Ajit's lab showed that in liver, NK cells are responsible for maintaining disseminated tumor cell in a dormant state. And this is mainly through secretion of interferon gamma. IL-15 help this process by inducing proliferation of NK cells. Here there's a dark side guy, bad guy called haptic stellate cells. And these cells secrete CXL12, which do the exact opposite things. They put NK cells to sleep, but induce cancer cell proliferation that lead to the termination of dormancy. So this is an example of a tissue-specific immune microenvironment dictate the cell fate of cancer cells. More recently, there's a back-to-back papers published on Nature Cancer by Dr. Yibing Kan's lab, documenting the role of complex formed by MTDH, MET adherent, and SND1 in antigen presentation and therapy. MTDH is long recognized to have the role in breast cancer metastasis by Dr. Yibing Kan's previous work. They interact with SND1, which is a nuclease. And the two together, they cleave RNAs, and including the type 1, type 2 messenger RNA, part of the antigen presentation machinery. And as such, the tumors can escape immunosurveillance, and they are not response to immunotherapies. But using a new compound, they can target this complex, to restore the sensitivity of tumor to anti-PD-1 treatment. In the final slide of this session, I'd like to put side-by-side two papers related to the same immune cell neutrophil. On the left, you are looking at neutrophil paper from Dr. Becker's lab, suggesting neutrophil can kill cancer cells by secreting the elastase. However, many cancer cells can escape this killing because they express elastase inhibitor, and this is happening more in murine system, as pointed out by the authors. On the right, you have another paper by Dr. Guohong Gu's lab suggesting that cathepsin C can be secreted by tumor cell to recruit neutrophil, and that in turn can promote tumor progression. So now we have a situation that same immune cell can both kill and promote tumor progression. And they have this is the opposite effect, probably because of different mechanisms that are at work at different contexts. And this direction may be determined by the cancer cell, but that definitely poses the question how to more accurately predict the immune microenvironment effects on tumor progression. Unfortunately, we still have a long way to go in this regard. To conclude this session, we know that cold tumors can be converted into hot ones. And many non-immune therapies actually work by activating anti-tumor immunity. And we can convert the bad immune cells into good immune cells. If we can recognize the mechanism underlying why they're bad. Also, tumor immune microenvironment might be organ-specific, as highlighted in the NK cell in the liver study. It also poses challenges. I would summarize the challenges that in my mind, into these two questions. If we can see there are multiple immunosuppressive components in the tumor environment, then how can we accurately identify which one is the key? Which one should we target? As the ecosystem is very complicated. And for same immune cell type, how can we distinguish the different functionality of it in a specific context? These require more research going forward. In the last couple of minutes, I'd like to use two studies to discuss the emerging preclinical models in breast cancer, or the emerging new utility of different preclinical models. The first is again from Dr. Alana Whelm's group, and Dr. Whelm has been systematically study patient-derived xenograft and patient-derived organoid models. Their previous and current work has demonstrated that these models can recapitulate some key features, including the profiles and also the therapeutic responses. But in this particular study, they demonstrate that PDOs can be used co-clinically, which means when the patient is receiving standard care, PDOs derived from the same patient tumor can already be subjected to drug screening in parallel, and therefore the best drug that can be used as a next line therapy will be identified. So when the standard therapy fails, and that drug can be immediately used. There's actually a real live patient example in this study showing the proof of principle that this can indeed improve survival. The second paper, which is also the last paper of this talk, is regarding prolactin, a new model called NSG Pro. And this is really useful because although we know prolactin is an important hormone in breast cancer biology, that we have had a hurdle to study it because mouse prolactin does not activate human prolactin receptor. And this group from Dr. Haiga Ri knock in human prolactin into the non-skin mice. And by such, they overcome this hurdle. And in this new model, they really show that ER-positive cell lines and PDXs are growing better, and they exhibit expected endocrine responses and resistance, and they also metastasize. So using these two studies, I'd like to convince you that PDX and PDOs are valuable platforms for co-clinical studies, and that there's a potential for them to be used for real-time precision medicine. And genetic engineered animal models continue to be useful if we can modulate them appropriately to reflect better human biology. There are a lot of unmet needs. For instance, for ER-positive tumors, we still lack immunocompetent model that recapitulate metastasis behavior as well as therapeutic responses. If you look at the literature, a large quantity of studies are still based on very limited choice of preclinical models. Although these are great models, they are unlikely to recapitulate the heterogeneity of breast cancer. Therefore, there's a need for a wider variety of experimental models used in metastasis research. I'd like to stop there by acknowledging all my lab members and my collaborators who contribute to this talk, the patient advocates who have supported our work relentlessly, and also funding agent. Thank you very much.